Okay, so we're on our final chapter, chapter 23. And chapter 23 is on biochemistry. I want to say a little thing about biochemistry. It is a wonderfully complex field of study. Many universities will have a biochemistry department that is entirely separate from the chemistry department. And we've got a physical sciences department, which uh, includes much more beyond chemistry. But I just want to make the point that biochemistry, deserves to be its own independent field of study apart from chemistry because of how unique it is from other branches of chemistry and how complicated it is and things. And I am not a biochemist. Now, reading over your textbook, I can tell that a good biochemist was involved in writing, and it goes into quite a bit more detail than the textbooks that I learned from. Uh, there's a lot in there that I would say, is not necessarily very important for you to learn right now. You'll be taking biochemistry from one of our two wonderful biochemists sometime in the future. And so, what I want to do is build a good foundation and give you a big picture about what biochemistry is, what are the important things about it, and whatnot, rather than trying to reproduce a whole biochemistry course in our last week of lectures here. Because biochemistry involves big molecules and complicated structures, I'm probably not even going to use the blackboard, so I'm just going to sit down here and put on our slideshow. And maybe I'll even point the camera down so you can see my head while I'm reading. Maybe that'll be weird, I don't know. Can you see my head? Not quite. Maybe if I sit up straight. I'll turn it down just a little bit more. Okay, that'll have to do. For the intro to this chapter, the first thing we're going to do is go over the four main categories of biomolecules. And what I think is maybe the most interesting thing here is not listing these categories and the functions that they perform in most organisms, but what's most interesting is actually how they defy being put into a box. So carbohydrates are usually said to be like energy storage molecules. If you're an athlete and you eat a lot of carbohydrates before your sporting event, like we call that carbo loading, and it's all about making sure that your body has enough energy to perform. But carbohydrates can really do so much more. First of all, I will use the board. I lied, but I need to make sure that I know where to write. Okay. So carbohydrates have the general formula of CH2O. That's their empirical formula. And so you've got a carbon and you've got like water. So carbo and hydrate, as in like hydration or anyway. So this is kind of the general empirical formula for carbohydrates. And then we'll see that larger and more complex carbohydrates maybe deviate a little bit from this precise chemical formula. But there's a lot of oxygen in a carbohydrate, and it doesn't have any other heteroatoms. It's all carbons, hydrogens, and oxygens. That's where they get their names. And so we can see here a couple of different categories. We'll talk about carbohydrates in more detail later today, but you've got simple sugars and then you've got polysaccharides, other more complex sugars. And they are obviously going to be providing energy. They also act as antigens, identifying blood and tissue types in mammals. So I believe oh no, this is where you'd be better to have a biochemist. But I believe your blood type, like if you're a type A or a type B or a type O blood, I think those are sugar markers on your blood cells. They could be proteins, but I believe those are sugar. Anyway, some antigens are sugars. Also, They can be part of the structures of cell walls in plants and fungi and bacteria and things. So carbohydrates are more than meets the eye. Okay, lipids. So lipids, you're probably familiar with lipids as being fats, fatty tissues and fatty molecules, and that is what a lipid is. And so fat, we typically think of as being an energy storage molecule in bodies. Of course, fats can also be an insulator. If you think of like a walrus or a polar bear, they've got a lot of fat that serves to insulate their bodies. But fats or lipids also can be communication molecules. Hormones provide chemical signals to govern and control really important biochemical processes. Phospholipids form the membranes of cells, and so they're a structural molecule. And then proteins, I think, protein is muscle. We think of protein as forming the structure. But proteins are also enzymes, so they're catalysts. They're involved in all sorts of chemical reactions, transporting oxygen. Collagen is the structure of skin. Immunoglobulins are part of the immune system. And there are some hormones which are peptides as well as the hormones which are lipids, and so they're involved in chemical signaling and communication. And then lastly, nucleic acids. So nucleic acids, the most famous of which is DNA, deoxyribonucleic acid, which stores information. And we'll actually have our last day, we'll talk about DNA and the various types of RNA, but they don't just store information. Nucleic acids are actually a part of the ribosomes, which will transpose that information into synthesizing actual proteins and things. And so DNA is really kind of involved on several levels of protein synthesis. And of course, DNA itself is a sugar, deoxyribonucleic acid. The ribo part of that is sugar backbone. So even DNA is made of sugar. Anyway, give me a sec here. I want to see how many. We'll get to the next figure. Oops, wrong one. There it is. Okay, so a lot of these molecules are, large biomolecules are going to be formed through a dehydration reaction. And we've seen two examples of those already, forming ester bonds and forming amide bonds. And then actually also forming ether bonds. Forming ethers also is a dehydration or a condensation reaction. And so what you'll have is two oxygen atoms. One of them is an OH and one of them is an oxygen. Oh, or they're both OHs. I'm so sorry. One of them is an OH, and then one of them is either an NH or an OH. Right? And then the hydrogen will join the OH over here to form water as a byproduct, and then the nitrogen or the oxygen here will bind to the carbon on the other side. So these are ester or amide bonds, and they're a big part of how biomolecules are all formed. Okay, so the next section is about sugars. So let me pull up the figure. So these are what we call monosaccharides. Saccharide means sugar. And there are a great variety of different types of sugars. The monosaccharides are made up of one sugar unit. And we see only 3 examples here, but there are a lot of them. And the sugar units we're going to want to characterize in 2 different ways. So the first way is let me talk first about the general structure of sugars. You'll notice there is one oxygen on every one of the carbons. And most of those oxygens are single bonded to the carbon and then have an H. And so you could think of this as like an alcohol. It's a hexol or a pentol. It's got like 5 alcohol groups. And then there's going to be one of the oxygens which is double bonded. So that's what makes sugar be sugar. Of course, I can eat sugar and I'm not going to get drunk off of it. It's not really an alcohol in the sense of methanol or ethanol. The fact that it has so many OH groups is going to be really important to how it functions in the body and things. And so it's really wrong to say that sugar is just an alcohol. It's very different than alcohols. Anyway, sugars are sugars. They are their own thing. Okay, so they've got OH groups everywhere, and then they've got one carbonyl group. So the first way we're going to classify sugars is by the number of carbons. These here all have 6 carbons, and sugars are always going to end in the name O-S-E. Os means sugar, and so sugar with 6 carbons would be a hexose. 5 carbons would be a pentose. 4 carbons, here we're going to deviate from our ordinary organic molecule naming. 4 carbons is a tetrose, and 3 carbons is a triose. And so sugars can have 3, 4, 5, or 6, or even more carbons, although the 6 carbon sugars are by far the most common. So here we have glucose, galactose, and fructose. You've probably heard of all 3 of those. These are hexoses. The next way that we characterize sugars is by the position of their carbonyl group. They'll always be either on the terminal carbon, making it sort of like an aldehyde, and those are called aldoses. Or they'll be on the second carbon, and that would be a ketose, because it's like a ketone. And so these first 2, glucose and galactose, are both aldohexoses. And then the last one there, fructose, is a ketohexose. And so what's the difference between galactose and glucose? They're both hexoses, they're both aldoses, and so they're both aldohexoses. What's the difference? And if you look at it, the only difference is in that 3rd carbon, the one that's kind of right in the middle of this chain. Glucose has the OH going off to the right, and galactose has the OH going off to the left. And so that's the difference between these 2 sugars. And then fructose, of course, is the ketose. So let's actually do a bit of a practice problem here. Okay, so can we classify these monosaccharides by the functional group and the number of atoms? So this first one, A, we're going to count how many carbons. There's 5 carbons. The 2 on the end have sort of a condensed structure instead of showing the hydrogen going off one direction and the oxygen going off the other direction. And the reason is because there's free rotation around that bond, and so it doesn't really matter. All the others, there is a difference between left and right. Anyway, so there are 5 carbons here. That makes it a pentose. And then the carbonyl group is on that second carbon. So this is a ketopentose. Number B, or letter B, has only 4 carbons, and the carbonyl group is on the end. So this is an aldotetrose. And then C has 1, 2, 3, 4, 5, 6 carbons. Carbonyl's on the end, so that is an aldohexose. So ketopentose, aldotetrose, and aldohexose. I don't want to get into it too much, but the differences between these, like the galactose and the glucose, for example, they are optical isomers of each other. There's a lot of optical centers of chirality, and so it gets kind of complicated, and we can define which two are actually left-hand, right-hand versions of each other, and which ones are kind of different in other ways. Anyway, so we're not going to get into that. But here's the next thing I want to talk about. I should just leave the slideshow up, huh? You can see me going through the slides. So sugars, when they are in water, will spontaneously cyclize. And I can't remember, I think it's like 75% of the sugars will be in cyclic form, and some minority will be in the straight chain form when they're in water. And so the way that they'll cyclize is through this formation of it's like an ether. They call it a glycos. Oh, hold up. No, they don't. So the double bonded, the carbonyl oxygen is going to form a bond with the carbonyl carbon is going to form a bond with one of the alcohol groups that is on one of the other carbons. And they'll tend to form either 6-sided or 5-sided rings. And when they do cyclize, because there is free rotation and they can be oriented different ways before the cyclization occurs, you can have one sugar that will form two different cyclic versions. So we see here an alpha form has the OH pointing down, and when we say down, we mean on the opposite side to that CH2OH group that is up at the top. That CH2OH group is like our anchor and our reference, and so alpha form has it on the opposite side. We could say it's trans maybe to the CH2OH group, and then the beta form has the OH on the first carbon on the same side as that extra CH2OH. And so this is how an aldose is going to cyclize. And it's got that one CH2OH tail and forms alpha and beta versions. And then fructose is a ketose. And it will cyclize in much the same way. The carbonyl is going to break its double bond with the oxygen so that it's just a single bond, and then sort of transfer that bond over to bond to the oxygen on carbon 5 instead. Only this time, because that carbonyl is on the second carbon, we form a 5-membered ring, and we have two CH2OH groups. One of them on the 5th carbon, just like before, and then the other one on that carbonyl carbon. So anyway, these also have an alpha and beta form, and that's going to still reference whether the OH group is on the opposite side from carbon 5's methyl group, or whether the OH group is on the same or different side. So this is how fructose will cyclize. So let's I think the best thing, because I can't draw this stuff on the board, so maybe the best thing is to just keep working our way through the slideshow. If we look at these monosaccharides, are they hexoses or pentoses, and are they alpha or beta? So they've conveniently numbered the carbons for us. We'll start with the carbon on the right side. That's the carbon which was the carbonyl carbon. And so this is 1, 2, 3, 4, 5, 6. It is a hexose. Furthermore, we could say that this would be an aldose, so it's an aldohexose because it is bonded through the first carbon there. And is it in alpha or beta form? We see that the OH group is up on top of the ring, just like the CH2OH group coming up off of carbon 5. So this is a beta form. And then the next one, we can count 1, 2, 3, 4, 5 carbons. Again, this is an aldose, so it's an aldopentose. And again, it is beta form. So this is a beta hexose and a beta pentose. So the next group are disaccharides. Here we see glucose and fructose, and when they have cyclized, they can further form a bond between individual sugar molecules, and that's called a glycosidic bond. And that's going to take two sugar monomers and form what we call a disaccharide. So this is sucrose. This is table sugar. Table sugar is actually not one of our hexose simple sugars. It's a disaccharide, glucose and fructose. And there's a lot of ways that we can describe exactly how that glycosidic bond occurs. If we let's see, that's sucrose. If we look at maltose here, for example, we need to identify which two carbons are going to form the bond. And again, we'll always number these cyclized sucrose molecules from the carbon which was a carbonyl carbon before it cyclized. And by convention, that's always the carbon on the far right. So you'll notice they both start one on the right, 1, 2, 3, 4, 5, and 6. Now, maltose is a disaccharide which forms by a bond between the carbon 1 glucose on the left side and the carbon 4 of the glucose on the right side. And it is an alpha linkage because that linkage occurs on the opposite side of the molecule from the CH2OH group. And so that is an alpha 1,4 glycosidic bond between two glucoses. That is the definition of maltose. You can see it gets fantastically complicated. Here we've got, I don't know what those sugars are, but they're two hexoses. One of them on the top, if we count those carbons, 1, 2, 3, 4, 5, 6, we'll notice that the cyclization occurred on the first carbon, so that is an aldose. That's glucose. I see the name now. And then the one on the bottom is fructose, and that one is a ketose. You'll notice that it has that CH2OH down below where the carbonyl carbon is. So this is a 1,1 alpha, I believe. Oh, no, I get it. Sorry. Because we're going to count the fructose for its first carbon is, of course, not the one with the carbonyl because it is a ketose. Okay, and then then that OH is on the same side as the fructose's CH2OH. Is on the far side from where the carbonyl carbon was. Okay, and so that makes the fructose is a beta cyclic fructose, and the glucose is an alpha cyclic glucose, and we're forming the glycosidic bond from the glucose is 1 to the fructose is 2, so it's alpha 1 beta 2. Okay, so the last one that I want to talk about, sorry, let me check and make sure we still have the slideshow showing. So our oligosaccharides. So we've got monosaccharides, glucose, fructose, galactose, and this just table just kind of shows you where they're found in nature, right? And we've got the disaccharides, things like maltose and sucrose and lactose are also all actually very familiar to us. Some people are unable to digest lactose, which is the sugar that is in milk products. Sucrose is table sugar. Maltose is kind of starchy, and if you've ever had malted milk balls or a malt shake or something like that, it's that form of sugar that is used to form those. So those are disaccharides. They have 2 sugar units, but you also have polysaccharides, which contain a lot of glycosidic bonds, hundreds perhaps. And we're going to talk about 3 different categories: cellulose and starch, which includes amylose and amylopectin, and glycogen. And they differ a little bit in their size and in the ways that they link together. They have very different physical properties and different functions in living organisms, but in the end they're all just sugars put together. So cellulose is used in plants to provide structure. Cellulose is actually one of the major components of cell walls, and they make up like wood, the main structure of plants. And so it's linear, and its linkage is beta 1,4. Okay, and it's just 300 to 3,000 monosaccharides, all beta 1,4 linked together. That was just like we saw with sucrose, was beta 1,4 linked, wasn't it? But cellulose would be, instead of just 2, it would be a bunch of them going on forever and ever. Amylose is as a starch, so amylose is like what a potato is made of. And it also has 250 to 4,000, so hundreds to thousands of sugar units. It's linear, meaning that it just has one sugar right after the other in a straight line, but its linkage is alpha 1,4. Then amylopectin is much larger than the others, 2,000 to 200,000 glucose units, and it's branched, and so we'll show you pictures of these in a minute. And then glycogen, this is where we store sugar in our liver, is in the form of glycogen, and it's a highly branched molecule, and you see it's up to about a million glycose units, so glycogen is huge. So this is what the cellulose structure looks like. It's rigid and it's long and straight, and that's what makes wood behave in the way that it does as a hard, dense material. That is cellulose. And even though it is made of sugar, our bodies are incapable of breaking those glycosidic bonds. And so I could eat sawdust all day long and die of starvation, even though it's made up of glucose. Its chemical formula and its chemical makeup to the untrained eye is identical to a potato, but I can't live off of sawdust. Now there are microorganisms and of course termites, which have a special enzyme that's able to cleave those glycosidic bonds and metabolize the sugar units one at a time. But we humans and most other animals cannot. So here's amylose on the bottom. It's just got alpha beta linkages. It's got alpha linkages instead of beta linkages, and that just is something that we are able to digest. It's not like it's just almost kind of a fluke of nature, that we do not have that enzyme in our bodies, but to digest cellulose. But amylose we do. Okay, and then amylopectin, you see it's like an amylose, but every now and again that 6 carbon is going to form a bond to a new branch. So that's amylopectin. Oops, wrong key. Oops. Oh, and they don't show glycogen. Glycogen would be like amylopectin, but just with a lot more branches. Okay. And so I know this is kind of brief. I'm not too concerned about making sure that you are gonna know all the ins and outs of oligosaccharides, which means things made up of more than one sugar unit. What I think is most important is identifying altoses and ketoses, counting the number of carbons, whether something is alpha or beta, and then identifying the properties of cellulose versus amylose versus glycogen. The linkages can get pretty complicated, and you saw I even struggled a little bit because you really have to go slow and pay close attention. And anyway, so I wouldn't worry so much about the links in polysaccharides. Just make sure you understand simple sugars or monosaccharides, how to identify and name them and categorize them. So we'll take one section a day from here on out, and I think that will give us plenty of time to not feel rushed through this last chapter and still have kind of short days, because I know you've got your midterm that you're probably focused on, and finish just right in time for the semester. So.